Hi, can somebody communicate? I got an error with my Zoom, so
I can hear you.
great. And I can hear you. All right. Sorry for being a bit late. I was logging into my Clark um, Canvas page, and I forgot that there's no link anymore to Zoom in the Clark Canvas page. So I had to log into Zoom, and to do that, it has verification who I am, and so that went through the verification process actually twice. Uh, so I'm a little late, just a little late. All right, are there any questions before we begin? I don't have that there. Come on. Let me state to begin with that if you come to a web page where it doesn't open in Zoom, it's because the page is trying to go outside of Zoom. Where do I have that? My YouTube site. There we go. So let me show this. Share my screen. So when you click on my YouTube site, for example, this link here, it will give you this message. Firefox cannot open this page. It's because Canvas doesn't want to open a page in Canvas that is outside of Canvas. But it's got a link here, my YouTube site. You click on that link, and then it'll open the page in another tab. Any question about that? And that will happen throughout the term because Canvas will not want to open a page outside of Canvas inside Canvas. And this is one that I know of because a student asked me about it. Let's go back here. And I was trying to open up my syllabus and I'm having trouble getting it because normally I just open up Word and it's the document there. And I did a lot of updating the website, so there's a lot of documents there. Ah, there it is. So let me remind you, today is July 11th, Thursday, and we're supposed to be talking about chapter 1 and chapter 10 in the lecture. And in the lab, we'll be discussing lab 01 today. That'll be happening in the lab time. Any questions about what we're doing? That's the wrong, yeah, that's the wrong, that was the wrong, I thought I got summer 2024. I got spring 2024. It's the same topic, but um, obviously I opened up the wrong syllabus. There we go. All right, let's move on to chapter 1. Let me open up a little bit of light in here so I can see me better. Let's go to where we left off. So I have that we had discussed biotechnology and that it was actually centuries old. A new aspect of it is genetic engineering, where you put genes in organisms, and that's a new aspect of biotechnology. So let's talk a little bit about microbial ecology. Microbial ecology is actually good for both the planet as well as humans. bacteria and microbes are needed to recycle carbon, nitrogen, nutrients, sulfur, phosphorus, and other elements that can be reused by plants and animals. If that didn't happen, and an animal or a plant were to die in the forest, for example, and those nutrients wouldn't be recycled, they would just sit there in the forest, never being used again. And we need the microbes to break down those nutrients and elements so that they can be reused by the ecosystem. These are examples of microbial mediated biochemical cycles that support life on Earth. And we've already discussed the nitrogen cycle, where that cycle really runs because of microbes. It is true that microbes help the carbon cycle and the sulfur cycle and the phosphorus cycle, but they do not run nearly every step of these cycles, meaning, well, like sulfur, it erodes out of rocks and then becomes available. But microbes can change the sulfur from one form to another. Like humans cannot consume sulfate or any other, I think, inorganic form of sulfur, but we do need sulfur, and we get it in a what's the word I'm looking for? In a organic form, like sulfur in amino acids or protein. The microbial mediated biochemical cycles supporting life on Earth were first discovered in the 1880s and to 1892. You don't need to know the time, by Winogradsky and Bajorink. And they were the first to show the recycling of nutrients. They first found it in nitrogen and I think sulfur, but then went on to find it in a few other cycles too. And the reason why they were able to find it was they were unique, and that is they didn't study microbes by purifying one species and then studying that species. They were looking at microbial communities of many different species living together. And they were the first to show the microbial mediated biochemical cycles like the nitrogen cycle. And if they had only studied one species, they would not have seen the nitrogen cycle. The most they would have noticed would be nitrogen being changed from one form to another. They would have never found the nitrogen cycle. And that's because you need to have many different species to get the entire cycle. Any question about any of that? Microbes are important in human welfare because they can break down, well, pollution and other nutrients. Microbes are important in bioremediation, life, and to remedy. We use living organisms to deal with environmental problems. That's why these gentlemen are spraying microbes on this oil spill. So the microbes will consume the oil and then break it down into CO2 and water. We use microbes, obviously, to break down sewage when we flush stuff down our sinks and our toilets. Well, bacteria can deal with pollutants, such as this oil spill, and even mercury. Now, microbes cannot break down mercury because it's an element, but microbes can convert this mercury pollution from elemental mercury, which is highly toxic, to an organic form of mercury, which is less toxic to us. So it's still toxic to us, but it breaks it down into a form which is less toxic to us. And then oil can be totally broken down from oil down into carbon dioxide and water. Any questions about any of that? Microbes can be used for biological insecticides. Many microbes are actually pathogenic to insects, and there are alternatives to chemical pesticides. For one thing, they're less toxic to humans and animals than man-made pesticides. And we use microbes to prevent insect damage to crops and prevent disease transmission. For example, Bacillus thuringiensis is a bacteria which can infect insects and be fatal to many insects. And yet Bacillus thuringiensis is harmless to humans and other animals and plants. My father in the 1960s actually put Bacillus thuringiensis on his apple trees, hoping to get no worms on the apples. And it did work in the sense that the apples had less worms than they would have if he hadn't treated them with Bacillus thuringiensis. But my father was way too lazy, and he only treated the apple tree once with Bacillus thuringiensis. And you need to treat the apple tree with Bacillus thuringiensis about every 2 weeks, the same as you do for man-made insecticides, to keep the apples from getting wormy. And then the other problem is my father was putting it on apple trees in Western Oregon, which is very much like Western Washington. And that is the rain would come and then wash down the Bacillus thuringiensis. And so what you need to do for getting wormless apples is you treat the apples every 2 weeks and every time after it rains. Any question about any of that? But if you would do that, you would get Bacillus thuringiensis working on your apples the same as a man-made insecticide, except it would be less toxic to humans. I'm not sure what organic apple farmers use, but that would be one thing they could use to get the apples organic and then worm-free. If you don't know and you're trying to grow apples, they will become wormy, and that's because there's a fly that they'll put an egg on the apple, and then it'll grow into a worm and eat the apple. And that's going to be true of all apples. Not all apples on the tree, but probably every apple tree would get some worms in the apples if you didn't treat them. All right, any questions about any of that? That's telling you a little bit about the importance of microbes and human welfare, as well as welfare for the planet. If there's no questions, let's move on to the naming and classifying of microorganisms. This was established, both the naming and the classifying of organisms, by this gentleman here. Does anybody know who this is? Who started the scientific naming system, as well as the scientific classification system? You should know that from your introductory biology 160 class.
Linus, I don't know how to pronounce it.
Yeah, it's the Latinized word, which he liked Latin so much he actually Latinized his name, and it's Carolus Linnaeus. If you Latinize his name, it's Carl Linnaeus. If you don't Latinize his name, and I might not be pronouncing that right because I think it was Swedish and I don't speak Swedish. But the book, surprisingly, kept his first name Carl, but then Latinized his last name, Linnaeus. And I don't know why they would do that. But anyways, Carolus Linnaeus or Carl Linné is the person who started the scientific naming system, which is still used today. His scientific naming system was so good that scientists adopted it, and we're still using it today. The classification system that he started is no longer used today because he classified all living things into plants and animals. And obviously, you know, there are more things than plants and animals. For one thing, microbes did not fit well into separating them into plants and animals. And I don't know what Carolus Linnaeus did. Probably things like a photosynthetic microbe he classified as a plant, and then a microbe that would go out and eat other things like Euglena, which actually moved. He probably classified it as an animal. But how he classified bacteria like E. coli, I just don't know because it doesn't fit easily into a plant or an animal. And he classified the fungi as plants because, well, they weren't animals, but they're not really plants. And hopefully you know that. So we don't use his classification scheme anymore because we have more things than just plants and animals. But we do use his scientific naming system. His scientific nomenclature, the scientific name, is also called the binomial name nomenclature because he gave every species two names. Each organism or each species has two names, a genus name and a specific epithet name. So for humans, our genus is Homo, and the specific epithet is sapiens, and then the species name is Homo sapiens. And this is called the scientific nomenclature or the binomial nomenclature because each species has two names. Any questions about any of that? And that system is still in use today. There are a few rules for the binomial nomenclature system, and that is all names are either italicized or underlined, meaning if I were to write a scientific name on the board, I can't write with italicizing, so I would underline it. But when I'm typing, I can italicize. The genus name is always capitalized, and the specific epithet is always in lowercase. All scientific names are Latinized, meaning they're based on Latin, and they're used worldwide. So, whatever language you speak, the scientific name for humans is Homo sapien. So, whatever language, we would always use Homo sapien to mean human. And I don't know what human would be in Spanish, which I suppose I should know because I took Spanish in high school, but I don't remember. And it's not human, but Homo sapien would be what you would use even if you spoke Spanish. The scientific name is either descriptive or honoring a scientist. Some examples, like Homo sapien, is descriptive. Homo means a man who, or being who walks on 2 feet, 2 legs, and sapien, meaning wise man. E. coli is both descriptive and honoring a scientist. Coli refers to the fact that it grows in your colon. And then Escherichia is honoring the scientist Esther, or Esther, I think it's Esther though, an early microbiologist. Staphylococcus aureus is descriptive. It literally means ball-shaped cocci growing in a cluster like grapes that's golden in color. Aureus means golden, and then Staphylococcus means ball like cell growing in a cluster like grapes. Any question about any of that? All right. After the first use of a scientific name, you may abbreviate the scientific name with the first letter of the genus, a period, and then the specific epithet. For example, Staphylococcus aureus should be spelled out the first time it's used, but after that you can use S period aureus. And then Escherichia coli should be used the first time it's used, and then after that you can abbreviate it E. coli. Both of these organisms are found in the human body, or it can be found in the human body, in or on the human body. Now, when I state that, let me say that E. coli is an exception, and even among scientists, E. coli is so commonly known as a name that even among scientists, sometimes the first time they write it, they will write it E. coli. Okay? And that's just because E. coli is better known than Escherichia coli. Other than that, you always use the full name the first time and then abbreviate. Any questions about any of that? All right, so that's the scientific naming. Let's turn to the classifying of microorganisms. As I mentioned, this was started by Carolus Linnaeus, but we no longer use his system. And before we classify microorganisms, we need to look at the classification of all living organisms on the planet Earth. How we classify all living organisms is we start with obviously all living organism, and then we classify them down into 3 domains, the 3-domain system. The 3-domain system is also called the tree of life because the branching sort of looks like a 3-trunk tree. Obviously, this would be what one branch would look like one tree looks like. But if you have a tree that had 3 trunks, that would be the 3-domain system. And what happens is you have one domain, and then it'll branch out into different smaller classification systems. What we're not going to talk about much. I will talk about the next one, and that's a kingdom. But beyond that, we're not going to talk about classes, orders, families, genus, and species, at least not now. We will talk about kingdom now, and later on we'll talk about families and genus and species, but not now. So the 3 domains are bacteria, archaea, also called archaebacteria, and domain eukarya. These domains are genetically defined, meaning we do define what a domain is. And how you get an organism to be in one domain is you sequence their DNA, and that will tell you what domain it belongs to, because all of the organisms in one domain will have a genetic sequence closer to any other organism in the domain than an organism outside that domain. Is that clear to everyone what I'm talking about? You sequence the DNA, and then every species in this domain will have the sequence of DNA more similar to any other species in this domain than any species outside that domain. So the domains are defined. That came as a surprise to scientists because they assumed the archaea and the bacteria were similar to each other. They were both prokaryotes, they were both single-celled organisms, but when we sequenced their DNA, we found that they are actually two different domains. And in reality, the archaea are slightly closer to the eukarya than they are to the bacteria. However, that closeness is very slight, that the archaea and the eukarya had a common ancestor. Let me blow this up, see if I can do that. They had a common ancestor for a little while, longer than the archaea and the bacteria, or the eukarya and the bacteria had a common ancestor. And it's trying to show you this one branched off first, and then these two continued. Let's see if I can blow that up even further. They had a common ancestor just for a little bit before they branched. And like I said, that closeness between these two is very slight. These two are slightly closer together to each other than they are to the bacteria. And that's very slight. All right, so after the three domain system, we have the classification of the kingdom. The definition for what a kingdom is and how many different kingdoms there are is in debate. And scientists have been debating that for at least 15 years now, and they've never come to a consensus. So, and they probably never will. Different scientists will give you a different, well, there is no real definition for a kingdom, but will give you the different numbers of kingdoms and what organism belongs in what kingdom. We will use the 6-kingdom approach, but realize there's a 3-kingdom approach, in which case the kingdom is the same as the domain. There's the 5-kingdom approach. There's the 6-kingdom approach, which we will use. And there's the 9-kingdom approach, the 15-kingdom approach, the 20-kingdom approach, and then higher order kingdoms approaches. I don't know how many there are, but something higher than 20 kingdoms. Any questions about any of that? All right, the approach we will use is 6 kingdoms, and that is the domain bacteria is its own kingdom bacteria. So the domain and the kingdom are the same. The domain archaea is its own kingdom also. So the domain and the kingdom are the same. The domain eukarya, though, is broken into 4 different kingdoms. Kingdom plantae, the plants, kingdom animalia, the animals, kingdom fungi, the fungus, and then kingdom protista, the protists. Any question about any of that? Now, let me state that the kingdom protista is not a monophyletic group because it is defined as a eukaryote that is not a plant, an animal, or a fungi. So all other eukaryotes go in the kingdom protista. And so it is not a monophyletic group. And that's what happens when you split the kingdom into 9 different kingdoms or 15 different kingdoms. You simply take the kingdom Protista and split it into more than one kingdom. Now, when you have the higher order kingdoms, like 20 or higher, you can start splitting the plants and the animals, maybe the fungi, I don't know. But the plants definitely are separated into at least 2 different kingdoms in the higher orders. You don't need to know that, but I'm just telling you that the main point is the kingdom is not defined. There's arguments about what a kingdom is and how many different kingdoms there are. And we're not going to get into that argument. We will simply use the 6 kingdom approach because it was the last kingdom approach that scientists agreed on. And it's also the approach that the majority of scientists use today, even though they will tell you that the exact number of kingdoms is in debate. Any question about any of that? All right. So let me state once again that when we sequence humans, Homo sapien, we will find our sequence of DNA more similar to plants. Let me blow this up. An amoeba, slime mold, then an amoeba sequence of DNA is to the archaea or the bacteria, meaning the sequence of DNA in amoeba is much more similar to our sequence of DNA than the sequence in the amoeba is to an archaea or a bacteria like E. coli. Any questions about any of that? All right. Now, your textbook says that the 3-domain system has a cell type, and there's only 2 cell types. There are prokaryote cells and there are eukaryote cells. Now, some authors don't call this a cell type, they call it a super kingdom. And that means it would be a classification above a domain. We won't use that because your textbook doesn't. But what you do is you have all living organism separated into a super kingdom, and that would be a prokaryote or eukaryote. And then you separate the super kingdom into the domains. And there's only 3 domains. And so what you do with the super kingdom is you separate the prokaryotes into the domains archaea and the domain bacteria. Your textbook author says that the bacteria have the cell type prokaryotes. The archaea also have the cell type prokaryotes or prokaryote cells. And then the eukarya, that domain, have the cell type eukaryote cells. Any question about any of that? All right, so let's talk about some of the characteristics for the different domains. The domain bacteria has cell type prokaryotes. They also have peptidoglycan in their cell wall. If a bacteria has a cell wall, it will have peptidoglycan present because peptidoglycan is the main molecule in the cell wall of bacteria. The bacteria multiply mostly by binary fission. For energy use, bacteria can use any possible energy source, an organic chemical like glucose, an inorganic chemical like nitrate, or photosynthesis, where they actually use sunlight to get their energy needs. Any question about any of that? All right, most of the bacteria are either free-living decomposers or free-living primary producers, but some of the bacteria are parasitic and some of the bacteria are pathogenic, meaning some of the bacteria can cause human disease. Any question about any of that? Let's move on to the domain Archaea. They have the cell type prokaryotic. Do the domain bacteria have peptidoglycan in their cells, in their cell wall? Anyone know? All right, no answer, so I will answer. The Archaea do not have peptidoglycan. So there's no peptidoglycan in the Archaea or in the eukaryotes. Some of the Archaea are found in extreme environments, and these are where we first found the Archaea, in extreme environment, like the methanogens living in methane, the extreme halophiles. These are high salt-loving organisms. The extreme thermophiles, organisms living at a warm, or I should say a hot temperature, like around 95 degrees Celsius. However, when we've looked further, we can find Archaea in all of the Earth's biomes. So they are everywhere, in all biomes. They make up a significant amount of the cells in the soil and in the ocean. They're not the majority, but they make up a significant amount. And these are not an extreme environment. Here's another question for you. Are any of the Archaea pathogenic? Can any member of the domain Archaea cause human disease? Anyone know? Think about it. I'm not going to answer that one, although you only have a 50-50 shot.
No, they don't.
No.
Yeah, they do not cause disease. The Archaea are not pathogenic. For this reason, and this reason alone, we will not talk much about the Archaea after this lesson. So this lesson, we will talk about the Archaea, but after this lesson, we won't talk much about the Archaea. And that's because the focus of this class is on microbes that cause disease. And none of the Archaea cause human disease, at least that we know of. Any questions about any of that? All right, so let's talk a little bit about the domain Eukarya. It can be split into 4 different kingdoms: the kingdom Plantae and Amelia, fungi, and Protista. The kingdom Eukarya, all the cells lack peptidoglycan. So if you ever find peptidoglycan, you know that that cell is a bacteria, because they have peptidoglycan in their cell walls. The kingdom fungi. Fungi is plural, the singular is fungus. You don't really need to know about chitin in the cell walls. You should know that they use organic chemicals for energy. So most fungi actually consume glucose or some related molecule, similar to humans consuming glucose or some related molecule for food. The fungi include multicellular molds, shown here, and then the unicellular yeast, shown here. The fungi also include the multicellular mushrooms, which the molds and the mushrooms do consist of mass of mycelia, and they're composed of filaments called hyphae, and we're looking at a hyphae right there, and that would be the mass of hyphae, mycelia. Even a mushroom has the hyphae, but the mushroom cells grow together to show you the mushroom. And I should state that what we're looking at is actually the fruiting body of the mushroom. The mushroom will have filaments underground, which serve sort of like roots. The yeasts, though, are all unicellular. Many of the yeasts are decomposers, but some of the yeasts are human pathogens, like Candida albicans, a human pathogen. It's also a part of our normal microbiota. So as long as it doesn't have a yeast bloom, it will be part of our normal microbiota. But it can cause human disease. We also have the kingdom Protista, the protists, and we can separate this kingdom into three different groups. The algae, which are plant-like protists, have cellulose in their cell wall, similar to plants. They engage in photosynthesis. That's why they're green, or many of them are green. Like the red algae obviously aren't green, but they're still engaged in photosynthesis. They produce molecular oxygen and organic compounds like glucose, which are important to animals, which cannot make glucose from CO2 and water. The protists also include the protozoa, the animal-like protists, and these include amoeba, paramecium, and euglena. These are called animal-like because they can move as well as they can consume organic matter or consume something by putting out a pseudopod and then surrounding what they want to eat and then bringing that inside the cell. The protista, the protozoa, can move by either cilia, flagella, or pseudopods. Remember that the prokaryotes, both the archaea and the bacteria, if they move, they can only move by having flagella, because that's the only movement structure that prokaryote cells have. But eukaryotes can move either by pseudopods, cilia, or flagella. Any question about any of that? All right, some of the protozoa are parasites and some can cause human disease. The kingdom Protista can also be split into a fungus-like protist. We're not going to talk much about them, but I will briefly mention that the water mold, which causes potato blight and actually caused potato blight during the Irish famine in I think it was the late 1800s, which brought a whole bunch of Irish over to America. It's actually a fungus-like protist. Any question about any of that? All right. So the kingdom plants can be separated into mosses, ferns, conifers, and flowering plants. All of the kingdom plants are multicellular. We won't talk much about plants in this course because none of the plants cause human disease. Plants are important, obviously, for us because we get a lot of our food from plants. And then the food we don't get from plants, we get from animals, which they get their food from plants one way or another. I mean, even if you have a fish which eats other fish, the baby fish eat the plant or photosynthetic organism. Let's move on to the kingdom Animalia. The animals include sponges, worms, and the scientific name for that is helminths, the insects, and the vertebrates. All of the kingdom members of the kingdom Animalia are multicellular. We will study the animals in a little better, greater depth because some of the animals can cause human disease, and the worms can be parasitic, and this can even cause infectious disease, a parasitic worm. Insects can sometimes bite people and then pass on disease, like yellow fever, for example, or ticks, which can be a parasite of humans or animals. Any question about the kingdom plants and the kingdom Animalia? All right. Let's return to microbiology and microorganisms and answer the question, what comes under the umbrella of a microorganism? And I'm going to say it's better stated as a umbrella of a microbe because we do have viruses and prions, which are microbes, but they are not an organism. And they do fall under what is a microbe. Anyways, a microbe will either be a bacteria, an archaea, a virus, or a prion, which these are special because they are not organisms. And then some of the eukarya. All members of protozoa are a microbe. Most molds and yeast, all yeasts are microbes. Most molds too. Some algae, if the algae is microscopic, meaning you have to look at it under the microscope, then it is a microbe. But the algae that is the brown kelp washing up on the Oregon or Washington beaches, that is not a microbe. And then the helminths, the worms, are also falling under the umbrella of a microbe. And you should wonder why the worms are considered a microbe if the adult worm is very large and you can see it with your human eyes, like the beef tapeworm. It can either grow up to 30 meters or 30 feet. Sorry, I forgot my unit there. I think it's 30 meters, but let me just check. Come on. Oh, good. Having trouble even getting there. I had it for a moment. There we go. So it's 30 meters long. Can be 30 meters long. So if they're 30 meters long, why do we call them a microbe? Anyone have a guess? Well, microbiologists study organisms that cause disease, and the worms can cause disease. So that's one reason. The other reason is that generally the life stage of the worm, which is infective in humans, is microscopic, meaning there are different stages of the worm, and some of those stages are microscopic, including usually, if not always, the stage of the worm which is infective. So for most worms, it would be a fertilized egg, which would be the infective stage, and that is microscopic. Even for the parasite, which can burrow through your skin and cause, what the heck is that? That's shigellosis. Anyways, cause disease. The parasite that's burrowing through your skin is microscopic. I suppose there are some. Well, no, they're not infective. There are some worms that some patients have, but the worm itself is not, the adult worm is not infective. You have to have the egg, which is infective. When we're talking about the helminths, you should realize that the helminths are not a monophyletic group. They are multicellular animals. Many of the helminths are parasitic. Some of the flatworms, some of the roundworms are parasitic. Like the cestodes are all parasitic, but that's a certain class of flatworms. But they're not strictly microorganisms, but their life cycle involves microscopic stages, and the microscopic stage is the stage which is infective in humans. Oh, I was starting to say the helminths are not a monophyletic group, and we call them worms because to us they look like a worm, but the worms actually are three different phyla. The annelids, like the earthworm, the flatworms, like the beef tapeworm, and then the roundworms, like the trying to think of a roundworm. What is that called? Pinworm, for example, is a roundworm. They are different phyla. And so they all look like worms to us, but when we sequence their DNA, they are as different from each other as they are to humans. So to lump them all together as a helminth, as what we call a worm, is actually a little mistaken, a little wrong, that they should be stated that they are three different worms and they belong to three different phyla, and they're not at all genetically similar to each other. They're as different from each other as they are to humans. Any question about that? All right. By the way, the annelids, like the earthworm, all the members of that family do not cause human disease. So all of the helminths that we would talk about would be a flatworm or a roundworm, because those are the ones that cause human disease. All right, let's talk briefly about viruses. We'll get back to talking about viruses late in the term. Viruses are acellular, meaning they do not have a cell. They do have a genome, and this genome can either be DNA or RNA, but never both. And the DNA or RNA genome is the core molecule of the virus. The core can be surrounded by well, it actually is surrounded by a protein coat. The coat may be enclosed in a lipid envelope around the protein coat. Not all viruses have an envelope, but some do. And viruses only replicate when they are in their host cell. Outside of their host cell, viruses cannot replicate. And in fact, they have little, if any, metabolism outside of their host cell. Viruses are not part of the three-domain system because they are not organisms. They do not have cells. Any question about any of that? All right, let's compare the three different domains, bacteria, archaea, and eukaryote with each other. The cell type of bacteria and archaea are prokaryotes. The cell type of the eukaryotes is a eukaryote cell. The chromosome of bacteria and archaea mostly are one and circular. The bacteria have no histone proteins in the chromosome and no introns. We'll talk a little bit more about histones and introns later in the term. The eukaryotes do not have one circular chromosome. They actually have many chromosomes and they are linear. A circular chromosome is one that is in a circle where the ending point is the same as the beginning point. In a eukaryote chromosome, like the chromosome X, it'll have a beginning point right here and then an ending point right there. And they'll be two different points. It's not a circle. Eukaryote chromosomes have histone proteins and introns associated with the chromosome. We'll talk more about histones and introns later in the term, but just note that the eukaryote chromosome differs from the chromosome of a bacteria, actually in every way, and that is there's one chromosome here, many there, circular chromosome here, linear there, no histones, no introns, and then histones and introns on the eukaryote chromosome. Any question about any of that? When we look at the ribosomes in bacteria and archaea have the same size, 70S, so those ribosomes are similar. The eukaryotes have a larger ribosome, and it's 80S. You don't need to know about the S, but that is the sedimentation coefficient where the ribosomes sediment or migrate to in an ultracentrifuge gradient. And that's just how chemists and molecular biologists classify ribosomes by their size. The cell walls differ. Bacteria have peptidoglycan in their cell wall. The archaea have no peptidoglycan in their cell or their cell wall. Eukaryotes also have no peptidoglycan in their cell wall. And as you know, animals don't have a cell wall, and all animal cells lack peptidoglycan. So this is one way that the bacteria differ from all others. Any question about any of that? All right, if not, let's move on to another topic, the history of microbiology. We'll only go over a brief history of microbiology, and you should know that microbiology originally dealt with the debate about the theory of spontaneous generation versus the theory of biogenesis, meaning could microbes in particular spontaneously generate. And then the history of microbiology includes the golden age of microbiology, early microbiology, whenever people looked and studied something, it was something new. And you don't need to know the time frame for the golden age of microbiology, but it was early in the history of microbiology. So there's the different steps of the history of microbiology, and there's the golden age, the step here. Now let's talk about the history of microbiology. When we look at the fossil record, we can find fossil ancestors of bacteria, which were the first life on Earth greater than 3.5 billion years ago. So equal to 3.5 or greater than 3.5 billion years ago. We have the first evidence of life on the planet. And it was actually the ancestors of bacteria today. And here you can actually see an actual fossil, and there's an artist's rendition of it. This is the only date you need to know for all of the history of microbiology that I'm going to give. So after this date, you can ignore the dates. I may state the date just to organize things. Okay? But you do not need to know any of the dates except for this first one. The first record we have of microbes comes from Babylonian clay tablets, 4,300 BC, where they wrote down recipes for making beer. And although the Babylonians did not know anything about microbes because they couldn't see them, they did make a recipe for making beer, which the beer is made fermented because of microbes falling in with the beer. And that's the first record we have of humans discussing a microbe. In the year 600 AD, the Mayans recorded the first time they made a fermented beverage from cacao, which is chocolate to us. You do not need to know the dates, but you should know that before the Mayans could record their making of fermented beverage that we call chocolate today, that the Babylonians had to record the first time they used microbes for making a product. And then that had to be obviously after the first fossil record of life on the planet Earth. Any questions about that? The point is you may need to know the relative order of things on a slide, but you do not need to know the dates. All right, Janssen would next be our in the history of microbiology. Janssen developed the compound microscope, and without his development or invention, we would never had microbiology. So Janssen first had to make the compound microscope. Surprisingly, scientists never looked at living tissues and then wrote about it until 1665, which you don't need to know the year, but that was, what is that? 75 years later, before people used a microscope to look at tissue. And Robert Hooke did that using the microscope to look at tissue and found little boxes making up the tissue that he called cells. Let me see if I can go through this. Nope, not there. What he was doing is looking at thin shavings of cork under the microscope. Let's go ahead and put this in. Put that in. We'll go ahead and separate this into two here. And what he found was cells. inside the cork. Cork is a bark of the cork tree, and you use this bark to make corks which plug bottles like a wine bottle. And why Robert Hooke called these cells is they reminded him of the room in a monastery, which we'll do that there. And there would be a door right here for getting into the room. And the monks called these cells. And Robert Hooke thought these looked like the monks' rooms, so he called them cells also. Any question about any of that? And that's where the term cell comes from. Robert Hooke saw cells obviously first in cork, but then he looked at other plants and found cells. When he looked at tissue to an animal, he didn't find cells. And it wasn't until 100 years or more that the microscope improved before people could look at tissue to animal and see cells. Why was Robert Hooke able to find cells in plants, but not in animals? What's the difference between plants and animals that you can see under the microscope? Anyone? Nobody's going to take a guess? Let me see if anyone's.
Would it be the cell wall?
Yes, cells have a cell wall, which is a very rigid and thick structure. And because of that, Robert Hooke was able to see the cell because of their cell wall. Animal cells lack a cell wall. They do have a cell membrane, but with a primitive microscope during Robert Hooke's time and for about 100 years after, the microscope was not good enough to see the cell membrane making up the cell. And that's just because the microscopes were blurry, and so no one could see the cell membrane. But Robert Hooke's seeing cells gave rise eventually to the theory that all living things are composed of cells. And that theory was actually written up and developed further by Virchow, and it's known as the cell theory, that all living things are made of cells. All right, all of the above are not specifically microbiology, but we had to have these events happening before we could have microbiology. Let's move on. The first observations of microbiology were begun by this man, Antony van Leeuwenhoek. And I'm not Dutch, so I may be butchering the pronunciation of his name. He was the first person to use a microscope to look at living things, and he described the first microbes. And what was interesting was wherever he looked, scrapings from his teeth, rainwater, peppercorn infusions, the water from the River Thames, which was feeding the city of London water, or giving the city of London water, he found microbes. The reason why he was the first person to see microbes, which you can kind of see him using this right there, was he was the best lens grinder of his day, and he made the best microscopes. Here's actually one of his microscopes. It was primitive by our standards, and it was also primitive because it only had one lens for magnification. So it is a simple microscope, not a compound microscope. but he was able to see microbes with his microscopes. And it was largely because he ground the best lenses and was able to make the best microscopes because he made his own lenses. He was able to see cells so small that they were not seen for about a century later by other microbiologists. And here is his drawing right here, the cells I'm talking about. And you can look and see that that cell, as well as that one, looks like a rod or a bacilli. That looks like a spirillum. And this one, figure E, that looks like cocci, two cocci right there. These are bacteria, and these cells were so small that nobody else saw them for another 100 years, meaning other people's microscopes were not good enough to see the bacteria. You need to improve the microscopes for 100 years before the microscope got good enough for someone to see a cell so small that it was bacteria. And that took 100 years after Antony van Leeuwenhoek first saw them. Any question about any of that? All right, let me go back. In the history of microbiology, there was, in the early history, a debate about how life originates. And there were two competing hypotheses to explain how life began. The one was spontaneous generation, that living things arise from a vital force coming into non-living matter and creating life. And the other theory is the theory of biogenesis, that life only originates from life. And everyone would agree that you had a baby and that only came about because two parents got together to make the baby. So there were some things that everyone agreed that life came only, or at least babies only came from biogenesis. But there were other cases where people said life spontaneously generated. Over a century ago, there were beliefs that when people threw a log into the fire, sometimes a newt would come out, and people would say, spontaneous generation of the newt. And then they saw flies emerging from manure. Once again, they argued spontaneous generation of the flies. The vital force came into the manure, converted the manure into the fly. And then maggots arising from decaying corpses. That was another argument for spontaneous generation. Most were logical, but none were scientific. Some were amusing, like someone actually wrote up that if you put sweaty underwear in a jar and then had in the bottom of the jar kernels of wheat, it would spontaneously give rise to mice. In reality, the mice climbed into the jar to eat and get the wheat, and then got stuck in the jar because they couldn't climb out. And the sweaty underwear actually had nothing to do with the occurrence of the mice. Any question about any of that? All right. So a brief sidetrack for why people argue that microbes might spontaneously generate is if you had a tube of media, it would initially be clear. But if you left the tube out, it'd initially be clear with no microbe growth in it. But if you let it have time and leave it out, meaning uncapped, it would eventually become turbid. And people use this as an argument that microbes could spontaneously generate. The first proof for or against spontaneous generation came from Francesco Redi, who you should know was also the first person to document, meaning write up, a controlled scientific experiment. What Francesco Redi did was he got 3 jars, left them open, put meat in them, and then maggots appeared in the meat. He then got 3 jars, completely sealed, well, put meat in them, completely sealed them, and found no maggots. The people who were arguing for spontaneous generation said, no, that's not going to work because you sealed the jar, you kept the vital force from coming into the meat, and that's why the meat had no maggots. But Francesco Redi was bright enough that he countered that argument, and that is he got 3 jars and he covered them with a fine wire metal net so the vital force could come into the meat, but he still saw no maggots. After Francesco Redi's day, it was accepted that a complex organism, such as a maggot, could not arise spontaneously. They needed to come from some other life, meaning a fly came in and laid an egg on the meat, which gave rise to the maggot. Any question about any of that? So for big organisms, they did not spontaneously arise after Francesco Redi's experiment. You don't need to know when, but there's the date when he published that experiment and probably did the experiment this year. Well, the trouble was microbes were growing even if you covered the flask with a fine wire mesh. And even if you sealed the container, there sometimes was microbial growth inside the container. About this time, when they were still arguing about whether microbes spontaneously generated or not, Rudolf Virchow published his concept of the theory of biogenesis, meaning it was a theory long before he published it, but he actually wrote it up and then added to it. And even after he wrote his theory of biogenesis, that life arises only from life, the argument continued for about 3 more years. And whether microbes could spontaneously generate or not. What happened was in 1861, Louis Pasteur came and did an experiment proving or disproving the theory of spontaneous generation. Louis Pasteur disproved spontaneous generation, thereby confirming the theory of biogenesis or the hypothesis of biogenesis by doing a number of things. First, Louis Pasteur showed that microorganisms are present in the air, and if they fall into the tube of media, they will grow, and that is where the tube gets its growth. It's not from the theory of spontaneous generation. He also showed that microbes can be destroyed by heat. And then he made his ingenious and famous swan nut flax experiment, disproving the theory of spontaneous generation. What he did was he got a flask with a long neck, put media in it, sterilized, well, I guess I should say, heated the glass flask like that and bent it into a swan neck, and then heated the media to kill off the microbes inside the flask. And then this flask is hollow to the outside so that the vital force could come in from outside and come into the media. But he noticed that the media had no microbes in it unless you take the media and bring it down to here and then bring the media back. What was happening is the microbes from the air would fall down into the flask right there, the neck right here. But because of gravity, the microbes would not go up the hollow tube and then come into the flask. And if you ever go by the microbiology lab on the third floor of the STEM building, you can actually see an experiment where somebody did this. It's actually Antoni Roberto. So Roberto Antonio, I forget now. Roberto, he put a swan neck flask, sterilized it, and has it outside the window of the microbiology lab. And that's been there for at least 10 years, never becoming contaminated in the media inside the flask. Any questions about any of that? So after Louis Pasteur's work, everyone agreed that even microbes only come about from the theory of biogenesis, that microbes do not spontaneously generate. All right, following Louis Pasteur's work, there were a large number of discoveries, including the relationship between microbes and disease, and this became known as the germ theory of disease, that microbes can cause at least some diseases. This also led to immunity, that people have an immune system that can make a person immune to getting certain diseases. And an example of that would be if you were to get polio, something you don't want, by the way, your immune system would build up and then you would be immune to getting polio a second time. That might not be a best example, but it's the one that came to mind. I didn't want to talk about COVID-19 or the flu because obviously the variant can change and then you can get infected again. But when you get the flu, you will be immune to getting that flu, that same flu, again. The trouble is the flu will make different variants, and then you can get later variants of the flu. And it also gave rise to the antimicrobial drugs. Louis Pasteur's work gave rise to the fact that, well, microbes are causing disease. May we come up with drugs that can cure the patient of the microbes that are causing the disease and then cure the patient of disease. In 1850, Louis Pasteur developed the basic concept for aseptic technique, meaning you get a culture containing only one species, and that would be a pure culture. And then you transfer that culture using aseptic technique so that you do not introduce contaminants into your culture. Louis Pasteur did this by limiting dilution. So what he would do was he would have a sample in a liquid and he would dilute that making different dilutions, and then taking each of those dilutions and setting up 10 replica tubes of each dilution. And what he would look for is the dilution where only one tube out of 10 had growth in it. The other 9 had no growth in it. And Louis Pasteur correctly concluded that if one tube out of 10 tubes only had growth in it and the other 9 had no growth, the growth in that one tube was likely to be started by a single cell. And so the growth in that tube would be a pure culture. And Louis Pasteur was correct in assuming that. However, that technique of getting a pure culture was very difficult and time consuming to get a pure culture. Robert Koch developed a much easier technique for getting a pure culture. And what you do is you streak your culture, your microbes, out on solid media to get isolated colonies. And if you send the cells through 2 isolation events, meaning you get the cells through 2 separate isolated colonies, one after the other, meaning you have 2 plates and you streak out one plate, you get your isolated colonies, you take from one colony on that plate and you streak it out again on a second plate to get isolated colonies. If you take from one isolated colony on that second plate, those cells have gone through 2 colony isolation events. And if you do that, you will have a very high chance of getting a pure culture. It's higher than 95%. About the same time as Dr. Koch was developing his technique for getting a pure culture, someone in his lab, his technician, Mr. Hess, developed nutrient agar, a new media, solid media, for growing microbes. And it was very good for growing many microbes. And then about this time also is when Winogradsky and Bajering developed the concept of the biogeochemical cycles, where different microbes were recycling nitrogen and sulfur on the planet Earth, causing the nitrogen cycle and the sulfur cycle. Any question about any of that? All right, Pasteur then later showed that microbes are responsible for fermentation. He was brought into this by people in the beer industry who wanted him to tell them why the beer was going bad. And he discovered that microbes were causing it's not grape juice. What do you make beer out of? The stuff you make beer out of. I forget what is malt or whatever. The microbes were falling in and fermenting it to make the beer. And then he noticed with grape juice, microbes would get into it and ferment it and make it the wine. And then he noticed that the microbes were fermenting the sugar in at least grape juice into wine, and the sugar in the malt to make beer. But he noticed that microbes were converting the alcohol into acetic acid, which is vinegar, and that was spoiling the beer and the wine. And out of this work, that microbes were spoiling the beer and the wine and possibly other foods, making vinegar in the beer or the wine, spoiling it. People ask the question, might microbes do something similar to people and then cause disease. Louis Pasteur further went on to show that the spoilage bacteria in the beer and the wine could be killed off by heating the beer or the wine with the temperature not hot enough to evaporate the alcohol, or at least all of the alcohol in the wine, but hot enough to kill the spoilage bacteria. And that we call pasteurization, application of a high heat for a short time, which is used in a variety of foods to keep them safe, to keep them from spoiling. Does not sterilize the food. As you know, pasteurized milk has a shelf life. There are microbes in it, but there are no human pathogens in it. And all of the microbes which directly spoil the food are killed by the pasteurization. And that allows the shelf life of the pasteurized milk to be longer than the shelf life of unpasteurized milk. But it still has a shelf life. The milk will go bad because of the other microbes, which are not killed by the pasteurization. Any question about any of that? Anyways, the realization that fermentation happened because microbes affected the organic matter, and then other microbes could affect the alcohol, creating vinegar. Could microbes have a similar relationship with plants and animals? That was the question. And then what may microorganisms cause disease? This concept became known as the germ theory of disease. There was not one person who put the germ theory of disease forward. It was a concept that was developed slowly by many different people. Originally, it was an important concept because originally it was believed that poisonous vapors were responsible for causing infectious disease. Like when a person went traveling through the swamp and then got cholera later, the people, the physicians and whatnot, scientists as well, believed it was because the person breathed the poisonous vapors when going through the swamp, and that the cholera microorganisms happening to be in the swamp had nothing to do with it. Okay. But in reality, you're walking through the swamp, you're exposed to the cholera microorganism. You might breathe that in or consume it, I guess, while you're eating in the swamp. And then you get the disease cholera. So the poisonous vapors were not correct. And that is the microbes cause the disease. There were some people who showed that diseases appeared to be caused by germs. Ignaz Semmelweis, and I could be butchering his name, I think that's German or Austrian. He advocated for hand washing to prevent the transmission of postpartum or puerperal fever between OB-GYN patients. Semmelweis was in charge of the, what do you call that, the birthing area of the hospital, and he noticed that there was a disease that was much more common when doctors and nurses did not wash their hands. If they washed their hands before giving birth, the transmission of disease was much lower. Schimmelweiss actually lost his position because the president of the hospital was insulted that Schimmelweiss was recommending that the doctor wash their hands before handling patients or engaging in medical work. Joseph Lister also showed that disease was caused by microbes. He came up with a chemical disinfectant to prevent surgical wounds, greatly reducing the surgery-related infection. But the real person who finally proved the germ theory of disease, and note that he proved the germ theory of disease, he did not propose the germ theory of disease, was Robert Koch. What he did was he proved that the bacterium Bacillus anthracis causes the disease anthrax. And then he provided the experimental steps he used to prove that Bacillus anthracis causes the specific disease anthrax. And that, written up, was known as Koch's postulates. This proved the germ theory of disease. What he did was he simply infected an organism with Bacillus anthracis and then followed them over time, and the organism got anthrax. That's only the start of Koch's postulates. We'll talk later about Koch's postulates later in the term, and this is an important concept. We will talk about Koch's postulates at least 3 times this quarter, this term. So obviously it's something you can expect on your quizzes and your final. Any questions about any of that? All right, around this time, Edward Jenner came up with the first vaccine. What he did was he made a vaccine against the disease smallpox. What he did was he got somebody with cowpox. It was a disease that, like, milkmaids got from milking cows, and it was a much milder disease. And then he inoculated people with cowpox, and they might have gotten cowpox, but they were protected against smallpox. And Edward Jenner actually proved this by injecting 2 boys. One of them was his own son, and showed that when smallpox came to the region, that the boys were immune to getting smallpox. Unfortunately, the boys both died of some other cause. They didn't survive to adulthood, but they at least did not die from smallpox. They were immune to smallpox. However, nobody understood why Edward Jenner's vaccine was working or how to make another vaccine. And it was not until Louis Pasteur showed what was going on and how to make a vaccine that people understood why vaccines are working and how you can make another vaccine. And we won't go into it, but actually Louis Pasteur worked on a chicken disease. I think it was a chicken cancer that was caused by a virus, and he made a vaccine to this disease and then realized why the vaccine was working. And what happened with Edward Jenner's vaccine was that cowpox was a virus similar to smallpox. And when you inject the patient with cowpox, the immune system will respond to it, attacking the cowpox virus. And why the people were immune to smallpox is that some of the patient's response to cowpox cross-reacted to smallpox because smallpox was similar enough to cowpox that the immune response also acted against smallpox, making the patient immune to getting smallpox. And that's just an immune response. People make antibodies and T cells. We'll talk a little bit about that much later in the term to respond to different agents. And when those agents acted against cowpox, some of them, like some of the antibodies, cross-reacted and acted against smallpox, making the patient immune to getting smallpox. Vaccination is actually a word derived from vaca. Vaca is a word that means cow. And so vaccination comes from the first vaccine from Edward Jenner, where he used cowpox. The protection that's generated from a vaccine is called immunity. Any question about any of that? All right. How are we doing on the lesson here? I think I'll talk about this last slide and then we'll quit. The birth of modern chemotherapy. Chemotherapy is the treatment of disease with chemicals. Chemotherapeutic agents against infectious disease is called chemotherapy, although today chemotherapy is usually reserved to a drug applied against cancer. And we have other terms like antibiotics for a drug against a bacterial infection. A chemotherapeutic agent against an infectious disease can be from an antibiotic, and that is a chemical produced by a living organism, either a fungi or a bacteria, that inhibits or kills other microbes, or from a synthetic drug, and a drug that is man-made and then can be used against an infectious disease. So antibiotics here is the textbook definition of an antibiotic. For example, penicillin G is an antibiotic because it is made by a mold. However, ampicillin is a synthetic drug or a semi-synthetic drug because it is not totally made by another organism, meaning people for ampicillin, they first make penicillin G and then they chemically modify it to make ampicillin. And then there's something, I think it's carboxycillin, which is totally made in the lab by man. These are all members of the penicillin family. And by the textbook definition, only the first one, penicillin G, is an antibiotic. The others, although they're members of the penicillin family, would be a synthetic drug or a semi-synthetic drug. We won't use the textbook definition of an antibiotic because you'll find that that word usage, even among clinicians, and certainly among the common public, when we use the word antibiotic, we mean a drug that works against a bacteria causing a infectious disease. The first chemotherapeutic agent that was ever made, meaning the first drug when somebody started looking for drugs, was actually done by Paul Ehrlich, and he found a drug which treated syphilis in 1910. He called it a magic bullet. It was salvarsan, a synthetic arsenic drug, which was able to keep the patient alive and then treat their syphilis and kill off syphilis. It was not the first chemotherapeutic drug ever found that people used to treat disease. That was actually quinine from the bark of the cinchona or cinchona tree, which was used to treat malaria. And when do you think Europe first had this drug to treat malaria? About what time? Anyone have a guess? Just guess. You'll be wrong, of course, but that's all right. I don't expect anyone to know this. No one's going to take a guess. Well, the class is about to end, so I will state this drug was found in the mid 1600s in Europe. What happened was Jesuit missionaries in Peru noticed the Native Americans eating the bark of the cinchona tree to treat their malaria, and then noting that the treatment worked to cure malaria. And so the missionaries sent the bark of the cinchona tree to Europe in the mid 1600s. And who knows when the natives of Peru first discovered that. Anyone have any questions? If not, I'm going to stop here and we will continue there next time. I'll see you at 7 for the lab. All right.